Hi, everyone. Welcome back from spring break. I'm sure all of you feel the same way that I do, that it was way too short. But let's go ahead and get back into our discussion about the immune system. At least today we can talk about something a little bit more kind of fun and immediately interesting. So we'll talk a little bit about COVID and importantly have an understanding of how things like vaccines can play a really important role in our response to fighting the COVID pandemic. So before I started in with the lecture, I want to make sure that all of you are aware of where we are in the schedule. So here we are right after spring break. And we'll talk about pandemics, vaccines, COVID. We'll continue talking about that on Wednesday. And then Friday, there's no class because it's Kohio Day. Next week is our second exam. So I want to make sure that this is not a big shock to you that our second exam is next week. Hopefully, all of you have at least noticed this on the schedule leading up to spring break. But at least now we'll have Monday will be a review session for the exam, and then our second exam will be on Wednesday. It'll be the same format as the first exam. And then next week, Friday, is Good Friday. So, unfortunately, because of these 2 short weeks with these 2 Friday holidays, it's kind of compressed our calendar a little bit. And that's why this second exam kind of pops up where it does. Okay, but at least we will still have an opportunity to review on Monday. And then as a reminder, this week you do have a quiz due, and the quiz is on the material that we covered leading up to spring break. So chapters 10, as well as the stuff that we will talk about this week, chapter 11. All right. Let's recall what we have been discussing in terms of immune memory. We said that one of the really important features of our adaptive immune system is that not only can our adaptive immune system, our B cells and T cells, be mobilized to help us fight against a novel pathogen, something that our body has never seen before. Maybe it's something that the human race has not seen before. Right? So our ability to defend against it is not really adequate in terms of depending upon just our innate immune system, right? We are not pre-programmed to recognize certain novel pathogens. Because quite frankly, remember, pathogens like viruses and bacteria, they're evolving so much faster than we are. And so it's not realistic to imagine that at any given day, the human body has somehow pre-programmed in our genes the recognition of all these different types of possible pathogens, right? Because every day that comes along is the threat of new pathogens through their own set of mutations and changes. So we have to have our adaptive immune system that can come to our rescue, save our lives, and importantly, remember this battle that we were engaged in and that we hopefully won, right? Because if your adaptive system doesn't win over the pathogen, well, then, this whole thing about immune memory is kind of moot anyhow, right? Because, you're dead, right? So immune memory, though, allows us to be able to keep around in our system a small population of B cells, T cells, that sort of represent the best B cells and T cells that our body came up with when we had to fight this novel infection and when our immune system succeeded in beating it, right? So some of those B cells and T cells stick around because they have the knowledge or the memory of the particular shapes of antigens that proved to be so useful in our immune system finally being able to beat the infection. So let's look at this, right? We said that the first time that you are exposed to a novel pathogen, one that your innate system, unfortunately, is inadequate to deal with. So let's pretend, for example, that we have this kind of googly-eyed, drooling virus here, right? Looks pretty menacing. But the first time you are infected with this googly-eyed drooling virus, let us imagine that your innate response is not enough to deal with it. For whatever reason, your complement proteins are not sticking well. Different sort of preset receptors on your immune cells, like macrophages, are just not very good at being able to detect, opsonize, and neutralize this pathogen. As a result, right, the pathogen is able to kind of gain hold in your body, and this googly-eyed drooly virus begins to multiply, and you start getting very sick. Right? And so after the course of roughly a week or so, right, you're feeling pretty miserable, right? And you have fever, you have all kinds of physiological reactions that let you know that you're sick. And this is when your adaptive system has to be kind of woken up to come save you, right? So during this time is when you are now selecting for naive B cell clones, naive T cell clones that happen to have the right Ig shapes and T cell receptor shapes to be able to detect this pathogen and also detect the cells in your body that are being infected by the pathogen, right, in order to slow the spread and really gain an upper hand in eliminating it. So this is our primary response. The first time you're exposed to a novel pathogen that requires your immune system, your adaptive immune system, to wake up and do its thing to save you, right? This is your primary response. So over the course of a couple weeks, so after the first week, you're feeling miserable, right? You're hating life, you're binge watching Netflix, so maybe life isn't that bad, right? But the truth is you never really think that you're actually dying, but you absolutely are, right? You are absolutely dying, and you're counting on your adaptive immune system to wake up and do its thing. So by the second week, your antibody titer. So by now you've selected for B cells that are being encouraged to turn into plasma cells and release tons and tons of antibody. Right? Remember, at first, the quality of the antibody is not so good, right? Because really it's just a handful, relatively speaking, of naive B cells that just have close enough fit IgMs on their surface. Those are the ones that are selected. Some of them are told to turn into plasma cells, and they release IgM, right? But in your lymph nodes, you are improving the fit of the Ig, right, through hypermutation of B cells, and you are also selecting for T cells that can really help facilitate this effective adaptive response. So your antibody titer goes shooting up, right? And the level of effector T cells goes shooting up. So your cytotoxic T cells that have T cell receptor that recognize, different types of shapes that are presented by the googly-eyed virus here, right? So that the cells in your body that are succumbing to the virus and they're actually making more virus particles, they are busy presenting bits and pieces of the virus, small peptides from it on MHC, right? So that cytotoxic T cells can bind to those cells and eliminate those cells to stop the spread of the virus. And of course, you have lots of high quality antibodies now that are surrounding the virus, opsonizing it, and helping things like your complement proteins bind better. So the important thing to keep in mind here, right, is even though maybe by the end of the second week you're feeling way better, right? Typically, even if you think about the worst time that you had like the flu or something like that, I mean, all of you guys are young. You're probably in bed for a couple days at most. Maybe you threw up, maybe you didn't, right? But then after a few days, you're better. And so by the end of the second week, you're ready to go back to work or school or whatever, right? But the reality is, in the background in your body, the adaptive immune response is still sky high, right? Look at this. Your antibody titer remains very high for several weeks afterwards. Why is that? Because think about it this way. If you got this virus from riding the bus or being in the classroom or being in the office at work or whatever, right, and that's what made you sick and you went home and you were miserable and you're sick, once you get better and you go back on the bus or the classroom or back to work, right, it's not like magically the virus is no longer there, right? You're just going to breathe it in again, right? So evolutionarily, we evolve to anticipate that whatever this nasty virus is in our environment, um, even though we're better, we're still constantly breathing it in. So that's why maintaining that elevated titer of high quality antibodies, because by now you've gone through all the um, affinity maturation, hypermutation, and the B cells that are turning into plasma cells, their cookie recipe, right, for the antibodies is way better than the first batch of B cells that came in and were selected. Right? And so those are the high quality antibodies that are now flowing through your blood at this high elevated amount, so that, look at this, each one of these yellow arrows here represents the next time you're exposed to the virus, right? You are protected because right away when the virus enters your body, you have these neutralizing antibodies that can surround it and eliminate it before it has a chance to make you sick again, right? Interesting evolutionary adaptation. Importantly, as we go not just a few weeks out from this primary response, this initial infection, but maybe now months or even years, you still maintain a elevated level of those high quality antibodies in your blood. And these are, mainly in the form of IgGs circulating around, and you have memory plasma cells that know how to make those IgGs, so that if the googly-eyed virus comes back again, right away, you don't have to start from square one again. Remember, I told you, this is the importance of immune memory, because if this virus threatened to kill you the first time, right, and you were miserably sick, but fortunately your adaptive system came and saved you, right? Imagine if we didn't have any immune memory, and the next time you breathed this virus in, you had to go through that same whole awful process again. What if the next time you're not as lucky, right? So having some of those high quality plasma cells sticking around, memory B cells, and having this elevated titer of antibody in you means if the virus comes back, you don't start from square one again. You actually start from a really good spot, right? Which is the best quality antibodies that you came up with last time. So maybe a couple of years go by, and then one day, right? You have a secondary response because why? You see the virus again. The virus comes back and infects your body again, right? This might be several years later, but fortunately, you have the memory B cells that remember the virus, right? So right away, those cells start producing antibodies again, right? This time, though, look at your secondary response here, right? It is tremendous compared to the primary response. Oh, so I should say, right, the very first time you're exposed to this virus and you get sick, that's your primary response. Every other time in the future, we call it a secondary response. We don't bother calling it secondary, third, fourth, whatever, because the truth is, there's no real reason to keep track of how many times you're exposed to it, because from your secondary response onwards, what happens is that your immune system, right, really takes it seriously. Fortunately, or hopefully, it's fast enough to release those high quality antibodies and get those memory B cells and T cells moving that the googly-eyed virus actually doesn't have a chance to really make you sick again. Right? So that's why you can be exposed to it again and not even know it. Right? And that's kind of the beauty of the adaptive immune system. Right? If it works well, you never realize that you're exposed again to that thing that made you dreadfully sick, right, when you were a kid. Right? But because it's the second time you're exposed to it, or any subsequent time, your immune system really takes notice. Because why? That first time you're exposed to it, hey, you're exposed to all kinds of things all the time, right? You can't really expect that your immune memory has enough sort of space in it, right, to remember everything equally strongly, right? Because there are some things that maybe you will only be exposed to once, right? Maybe like when you're 6 years old or 16 years old, right? Or 26 years old or something like that. And maybe for the whole rest of your life, you never encounter that same pathogen again. That'd be kind of a not very efficient use of resources if your immune memory was just as strongly remembering that compared to some other virus or pathogen that you actually encounter like once every year or something for your entire life, right? So that's why the first time you're exposed to something and you mount a primary response, which is absolutely necessary to save your life, right? Your immune memory is kind of like, yeah, okay, we should remember this, but we're not really sure how important it is to remember this. And sure enough, over many years, if you never encounter that same thing again, right, your memory begins to fade because there's less and less opportunity to kind of exercise those memory cells, right? And over time, your body's just like, all right, forget it. But if you are exposed to that same googly-eyed, drooly pathogen again, right? Evolutionarily, your body says, oh, wait a minute, right? Clearly, this is something that persists in our environment. Clearly, this is something that threatens us. It wasn't just a one-time thing, right? Therefore, it's really worth it to take it seriously and invest more resources and more attention in making sure that we have a stronger immune memory now, right? So that's why, even if you don't feel sick the second time at all, because your memory cells did a good job of eliminating the virus before it had a chance to make you sick, your adaptive immune system in the background ramps up a big response, increasing not only the quantity, but the quality of these antibodies, right? And now the memory that you have from this point on is much stronger memory than you got just from your primary response. Okay. So let's think about this. Surviving a prior exposure to this nasty, googly, drooly virus yields secondary immunity, right? Meaning we have memory now to protect us the second time, third time, fourth time, whatever. Let's think about this cartoon. During the primary infection, let's say we had this aggressive form of the pathogen and it made you really sick, right? Fortunately, right, after binge watching Netflix and drinking lots of soup or something like that, you got better because your adaptive immune response came up with the B cells and the T cells that had the recognition, right, of the particular pathogen. Right. So you have T cells. This is our helper T cell, friendly helper T cell. He looks very friendly. This is our B cell, right, who generates antibodies. I don't know why they draw him like this. I think it's funny though. He's like squeezing out all the antibodies, right? But okay. And that's your protection. That's your memory. So that the next time exposure that happens maybe years later, this is our secondary response. Here's the googly-eyed drooly pathogen. He says, nice to meet you, right? But then you're like, no, we've met before, because you have these T cells and B cells ready to go. And as a result, the aggressive form of this pathogen does not make you sick again. And if anything, it strengthens your immune memory because now your immune system takes it very seriously, right? Think about this scenario, though. This is interesting. Viruses mutate all the time, right? So what if in reality, the version of the virus that you breathed in on the bus or at work, or your friend who coughs on you, great friend. What if in reality that virus this is kind of a funny, lazy virus. It was sort of mutated in such a way that it's really not as aggressive, right? So this is our lazy virus here, right? And so, it infects your body. It multiplies slowly in your tissues because it's lazy. It's kind of defective, right? But, it is infecting you and it is a virus. So even though it doesn't make you sick, or I should say maybe it doesn't make you as sick, but maybe you feel like a day of having a headache and a little slow, but then you're better, right? You still develop immune memory against it. And importantly, what? If the features of that virus, meaning the different three-dimensional shaped, proteins or other molecules that serve as antigens on its surface, if those things are still the same in the lazy version compared to the aggressive version, and that's what you select B cells and T cells to recognize, then what happens years later when the real aggressive form of the virus comes along, right? And he says, haha, nice to meet you, right? Once again, you have immune memory against him, right? Because your B cells and T cells already recognize those features. And therefore, the aggressive form of the pathogen does not make you sick. So this happens naturally. There might be the weak form of a pathogen that it might have mutated to become less aggressive. Or think about this way. You know that different viruses and bacteria belong to families, right? Because evolutionarily, they have similarities. They're related in such a way. So it could be that it's a different pathogen that infected you the first time, but it presented many of the same types of antigenic signals that this later really aggressive googly-eyed drooling virus presents when it infects you. Therefore, you may already have secondary immunity against this aggressive virus simply because you were exposed to that weakened form or different virus that still presented the same types of antigenic signals for your immune memory to learn. So hopefully you see where I'm going with this, right? Because this is the premise. This is the basis of vaccines. This idea that just as long as you show your immune system the key three-dimensional kind of surfaces, molecules, signals, antigens, right, whatever you want to call them, that you want your immune system to develop immunity against, right, then you may protect against the real pathogen when it comes and infects. So this is just a crash course history lesson in vaccines. So the grandfather of modern vaccines is Edward Jenner, who was a British physician. And so Jenner had learned that in many Far East places, Asia, India, it was a common practice in certain villages that when there was a particular, let's say, spread of a virus or some sort of illness going through the population, that oftentimes the village residents would go seek the advice of their sort of elder shaman or wise man medical provider. And there were stories that oftentimes what would happen is that the wise man would take somebody from the village who had recovered from something like, you know, smallpox, for example, right? And they would actually take bits of the sort of scar tissue, like scabs and things like that, from the person's skin, and then people would actually line up in front to visit the wise man, and he would basically put a little cut on people's skin, and then push or press or rub in some of this scab tissue from the recovered, fellow villager onto it, and that this process somehow gave the villagers protection against being infected, right? And so what was probably happening was in the scabs or the healing lesions of people who had recovered from things like smallpox were probably lazy versions of the cowpox virus, mutated versions of the virus that then, when they were on purpose introduced into one of the fellow villagers' bodies, right, the fellow villager's body could mount an immune defense against it, even though it didn't make them sick, right? That was the key, right? They would not get sick, but their immune system would see what this pathogen looks like, select for B cells, select for T cells, and build up a secondary immune memory against it, so that when the time came and the real smallpox came into their body, right, they were protected. So Jenner took this concept and he took it a step further, right? Because he realized that, you know, it's really dangerous to be actually taking smallpox virus on purpose and introducing it into people, right? Hoping that somehow they would be able to get immune memory without actually getting sick from the smallpox, right? Because the truth is, a lot of times people who got this type of a treatment actually developed smallpox, right? So Jenner made a very interesting kind of conceptual leap. He said, in other animals, there are diseases very similar to smallpox. For example, in cows, cows suffer from a type of pox disease called cowpox. Okay. And so what Jenner did was he took the cowpox form of the virus from, you know, cows who were suffering from cowpox, and he inoculated, right, or gave this to a little boy So the history books say that the very first person who was vaccinated was this little boy who Edward Jenner treated in this way. So Jenner scraped the little boy's arm, right? And he on purpose rubbed some of this tissue from the cowpox animal or the cow that was suffering from cowpox into the boy, right? And because it was cowpox, it was different enough than the human smallpox so that the boy did not get sick, right? But what was happening inside? The cowpox and the smallpox are similar enough that there are certain types of proteins, certain types of antigens on the surface represented by these different colorful shapes, right, that are common between them. Right? So that means in the little boy's body, right, he was exposed to the cowpox virus. And so the cowpox virus is kind of like a lazy version of a person's virus, right? It's not the correct type of virus to multiply out of control inside of the human body, right? But it does an okay job. So it begins to multiply a little bit just so that the body kind of like wakes up and realizes, we're being infected by a virus here. But fortunately, the cowpox multiplies slow enough that the little boy's adaptive immune response has enough time, right, to select for B cells, T cells, and destroy the cowpox. So as a result, in the little boy's immune system, he develops these different antibodies, right? Each one of them, the blue one, the red one, and the green one, represents what? A different naive B cell that boy happened to have floating around in his system that day, right? And each one of these then got selected, right, as a lottery winner, right? Maybe some of them better than others, but all of them got selected and all of them started being encouraged to go ahead and turn into plasma cells and turn into go up into the lymph nodes in the germinal centers and start to hypermutate and make better and better versions of these three antibodies. Right? So then what happens later is if this little boy is exposed to smallpox, right, the people version of this disease, you can see here what it's showing you is that 2 out of the 3 antigens on the cowpox are also shared by the smallpox. So this boy's immune memory already knows how to make the blue antibodies and the green antibodies, right? The red one turns out to be not really relevant against smallpox. So the boy's immune system was able to protect him against smallpox just because he previously was exposed to cowpox, right? And that's where the name vaccine comes from, right? Because this cowpox is a cow virus or vacca, right? Vaccinia virus. So that's the origin of the word vaccine. It comes from cows, right? So the principle here is that only the targeted antigens, for example, the blue one, this blue protein or whatever, or this green protein or whatever on the surface of the virus, only those things, right, really need to be presented to the immune system in order to develop adaptive memory against the entire pathogen. But you probably realize this, right? If this blue triangle and this green semicircle, if these are just like little proteins, right, or little peptides that come from little proteins, our blood is filled with all kinds of scrap pieces of proteins, random little fragments of things that are totally normal, right? So you can imagine then that just giving a person some of these little proteins, like the blue triangle and the green triangle alone, is not going to be enough to really get their immune system to take this seriously, right? And in fact, chances are your immune system's not even going to care about this because it just looks like, 2 small little random protein fragments in a sea of all kinds of otherwise normal protein fragments floating around, right? But the principle is there, though. The principle being that all you really care about is that the immune system has a chance to see these particular antigenic targets, right? Because that'll form the basis of selecting for an adaptive immune response. So that means vaccines, modern vaccines, there are many different approaches for making vaccines. Yes, you could just have a vaccine that is just the specific antigens that you want the adaptive system to learn about, right? But as we already discussed, those types of vaccines aren't terribly effective because you inject that into a person and great. So now you have some random proteins floating around in your blood, right? Your immune system, it doesn't care, right? More popular ways of doing it is to actually have the virus particle still be intact. So viruses that are intact and therefore have all of the antigens on their surface that you want your immune system to recognize, right? You can go ahead and sort of neutralize those viruses by heating them up, right? So that the proteins inside of them no longer function properly, or by chemically treating them so the virus no longer has replicative potential. So in effect, quote unquote, killing the virus, right? But I put killing in quotes because really, virus really isn't alive to start with, right? It's just some genetic material with some proteins wrapped up in a lipid coat, right? But the advantage of using a killed virus like this is there's no risk of infection, right? If you've destroyed it enough, even though it's intact, but it's no longer functioning, it's not going to somehow magically wake up and cause an infection, right? But again, the problem is, just like when we talked about, if you have individual little proteins floating around as antigens, having an intact virus particle, great, right? But if it's not doing anything, your immune system might not actually care about it either, right? So another way is to take viruses that are attenuated or weakened. So remember, in our slide earlier in this lecture, I used that example of the lazy googly-eyed virus, right, who mutated in some way that this virus doesn't replicate as quickly. So even though it still infects your cells and is able to create, right, a natural sort of infection taking place, which is what our immune system is geared up to recognize after hundreds of millions of years of evolving to be finely tuned to recognize the signs of cells being infected and viruses being made, right? We said that the googly-eyed one, because it's slow, right, it's able to still infect cells and cause your immune system to wake up, right? But because it's slow, your immune system can wake up before the virus has actually made you sick. Right? So you can do this artificially. Take a virus and modify it so that it is weakened. One way that is very common over the years is to take a human virus, the one that's actually making people sick, right, and force it to try to replicate in some other type of animal cells. An animal cell that's close enough to a human cell that this virus that is fine-tuned to replicate in a human cell can still do it in another cell. But because it's not really a human cell, it's kind of slow and whatever, right? And what you try to do is you give it plenty of time to kind of mutate so that it becomes better and better and more finely tuned to that type of an animal, right? Before you then have now this modified version of the virus that if you then put it into a person, right, no longer is as aggressive. Right? So that's what's pictured down here. Let's say this virus particle is an aggressive human virus. And so in the lab, you can grow them, right, using human cell lines to actually make a lot of virus particles, right? And all of these have the correct features on its surface. In this case, the little blue square represents the little blue, surface protrusion or glycoprotein spike that binds to a particular shaped, let's say, receptor protein on the human cell. That's what allows this cell to infect human cells so well, right? And then the human cell makes a lot more of these human viruses. So what people can do in the lab then is if you have researchers, for example, at the CDC, the Center for Disease Control, that want to create vaccines against this, they can take these human viruses that they're growing in the lab and making a lot of them, and they can infect things like monkey cells with them. So these blue cells are monkey cells. So they're close enough to people cells so that this virus that's geared up to multiply in people cells will still work. But you can see the difference here is the monkey cells, their version of the cell receptor protein, this blue thing, is a little different shaped, right? The cartoon shows it being rounded instead of squared, like in the people cell. So therefore, the virus, right, doesn't infect as well, right? This is the lazy part of it, right? But some of it gets in, right? And so the monkey cell then is hijacked and it makes more of this virus. But you keep doing this, right? You keep making this virus try to cultivate it or grow it in the monkey cells. And eventually what happens, right? You start selecting for certain random mutations that happen in the viral genome. So now the viral genome, instead of being a solid blue little, I don't know if this is meant to be RNA or DNA or something, right? It gets random mutations. And over time, you select for variations of this virus that have now, their glycoprotein spikes are now these yellow things, right, that fit the monkey cell surface proteins a lot better. And so they become better and better and better at infecting monkey cells. But in that process, what does that mean? That means they're getting worse and worse and worse at infecting the original human cells, right, that they came from. So now these attenuated or lazy versions of the virus can now be used as a vaccine, right? They can be introduced into the human body. Right? They are lazy, so they don't do a great job of infecting. They can still infect because, of course, you still want a little bit of infection to happen so that the natural signs of virus infection are happening inside of your body so that your immune system takes it seriously. But fortunately, because these are now lazy versions, right, your adaptive immune response has a chance to act quickly and learn from it and destroy it before they make you sick. So those of you who are kind of astute or paying attention here, you might say, yeah, but wait a minute. I thought the whole point was, now this thing has these yellow, glycoprotein spikes that don't work so well with people. But the thing that you're trying to protect against is this original virus, right? Yeah, I mean, the cartoon's a little oversimplified because, of course, just like in the previous slide I showed you, right, there wouldn't just be one particular antigen on the surface here that your body is able to identify different B cell clones against, right? Like in the smallpox, cowpox slide on the previous one, there was like a blue triangle, a green semicircle, and some other shape, right? So the idea is, even though certain things have changed on the surface to make this a lazy version of the virus, it still has many other features on its surface that haven't changed, right? That are still the same as the original human infecting version of the virus. And those are the things that your immune system will learn to recognize and select for and use to protect you when the real human virus gets in you. So what's the downside of this? The downside of this is because this virus only became lazy due to mutations, right? It's possible that inside of the body, as it is infecting cells, right, even though it's kind of lazy about it, hey, there's all kinds of mutations taking place there too. So it's possible, right, that some of these virus particles can actually back mutate or mutate in a way that restores their aggressiveness in people. And so that's why you can actually have cases where people actually develop the illness from the vaccine itself. Okay. And that historically is very important because poliovirus is a good example of that. The poliovirus was introduced in the 1950s at a time when it was very common for people to be infected with the poliovirus, right? And so I don't know if you guys have ever seen these old black and white grainy photos from the 1950s of entire hospital wards filled with kids who are paralyzed, right, in wheelchairs, whatever, because the poliovirus infected them and destroyed their nervous systems. Right, so thousands of people, tens of thousands of people, were impacted by this, and it was very common back then to know somebody who, has polio or has a kid who has polio. And then Jonas Salk introduced the polio vaccine, right? And the polio vaccine was based on an attenuated, lazy version of the polio virus, right? And it was super effective. Right? So due to a very aggressive vaccination campaign through the 50s and 60s, by the time the 90s rolled around, right, polio was pretty much eradicated. Right? We had pretty much eliminated it entirely from the population. But the irony is those few cases of polio that would pop up in the US in the early 90s, right, only came from people who were vaccinated and actually got polio from the vaccination, right? Because the attenuated virus, by unlucky chance, in their bodies mutated and became aggressive again and gave them polio, right? So for a while, there was a big outcry Because, hey, what are you doing if the only instances of polio are actually from people that you're giving polio through the vaccine, right? So that's why they changed the vaccine. And now the version of the polio vaccine that's actually inhaled, right, cannot infect you this way. It's based upon a different type of mechanism, okay? So mutations, viruses, bacteria, they're very prone to mutations. I mean, you think about a virus particle, right? A virus particle, as I said, is what? This teeny tiny little thing that's way smaller than even a cell in your body. And all it is a little bit of genetic material, DNA or RNA, with some proteins wrapped up in a little lipid bubble, right? And so if somebody coughs and you have this cloud of these little lipid bubbles, guess what? You know, the moment it's outside of the body and the sun's shining on it, you're going to have all kinds of random mutations taking place in the DNA. I mean, we worry about our skin cells getting mutations from sunlight shining down, right? So we wear sunscreen and stuff like that, right? You think our skin cells are subjected to mutations from the sun. Those are skin cells that are protected under thick layers of keratinized skin, right? So what chance does a teeny tiny virus particle have if all it is genetic material with some protein, some water, and a lipid bubble, right? So you better believe the moment you cough out a cloud of virus particles, so many of them are just getting zapped by the UV radiation from the sun to the point where their DNA doesn't work anymore or their RNA doesn't work anymore, right? But that means you have some where the mutations that take place are not enough to ruin the ability of the virus once it gets breathed in by somebody to still infect, but there might be mutations that take place, right, that might alter the way the virus functions. So the flu is like this. So when you have a local outbreak of something like the flu, that's what we call an epidemic. So the flu is a good example of an RNA virus. It's a retrovirus. Typically influenza, is highly contagious. We have other types of viruses like cold viruses, like rhinoviruses and adenoviruses, right? They all sort of behave a little bit differently, but the principle is the same, right? If, for example, you catch this flu virus and it makes you sick, let's say you are person P, all right? So you get sick, you caught it at school, you go home, you're laid up in bed for a few days, feeling miserable, hating life, right? And you develop, fortunately, right, a selection of antibodies that can recognize the virus particle. So here we have the virus particle, and this is, let's say this part of it here, this is the hemagglutinin spike that normally would bind to this particular, I don't know what this is, a sugar group that's on the surface of one of your cells. And that's how the virus would normally get in, right? But because you were able to choose and select for B cells that make this blue and this green antibody, right, they're targeting different epitopes or different three-dimensional features of the same antigen. So if we call this H, the hemagglutinin protein, glycoprotein spike, the antigen, right, on it are many three-dimensional features, each one of which could potentially be a good epitope for an antibody to be selected for. So in this case, the blue one, yeah, it's pretty good. It binds to this virus and it might help opsonize or get complement protein sticking on it. But really, right, the valuable one is this green one, because it binds over here and actually blocks the ability of the virus to bind to your cells. So as a result, hey, you feel a lot better, right? Hey, I should probably stay home a couple more days because I'm still coughing and stuff, but I feel way better. I'm going to go back to school, right? So you go back to school and you sit next to your best friend and your best friend says, oh, I'm glad you're feeling better. And you're like, yeah, I'm feeling way better. Thanks, right? And so as a result, you give the flu to your friend, right? And then she's person Q, right? So she goes home feeling sick after a few days. And what happens, right? The virus in her body mutates. So now you see the hemagglutinin part of the protein spike now has this like yellow part on it, right? So you're trying to pretend like the mutation happened in the DNA here or the RNA, whatever. And now the shape of the proteins a little bit different, right? This yellow part is slightly different shaped than the original red part. So she gets better, right? And she goes back to school, right? And then you see her and you're like, hey, I'm glad you feel better. I'm sorry I gave you the flu. And she's like, no, it's okay. Don't worry. I know you didn't mean it. All right. Right. And she coughs on you and she gives you this version of the virus, right? That unfortunately, even though you have the blue antibodies and the green antibodies, they don't work anymore against this. Well, maybe the blue one does, but but the green one was the one that was supposed to protect you. So guess what? You get sick from it again, right? Okay. So that's why it's very important to keep in mind that even after you feel better from something like the flu, be careful because you can still spread it to somebody else who may ironically end up having it all mixed up in their body and give you a version back again that you don't have immune memory against, right? So you take turns. Hey, but that's what friends are for, right? Take turns. Okay. So that's why highly mutable viruses like the flu, right, very easily escape immune memory. So let's imagine, for example, right, that the first time you're infected with the flu, let's say among the multitude of different antigenic molecules on its surface that your immune system could select antibodies against, let's say there are 4 important ones, 4 really good ones that you develop antibodies against, A, B, C, and D. Right? So your primary response the first time you're made sick by it, you're in bed for 14 days, right? Well, this is kind of extreme. I think they're trying to scare you, right? Because I don't know who would be sick for 14 days from catching the flu. I mean, at least if you're a college student, because you're like 20 years old, right? But anyhow, so you develop antibodies that recognize A, B, C, and D antigens on it, right? Let's say like a month later, all right, you are reinfected with it. And in this case, by now, in the population, the virus is mutating. So now it's A, B, C, and E, right? A different shape thing. So now the memory you have against the D antigen, right, is no longer useful. But fortunately, you still have immune memory against A, B, and C, right? So that's good enough to really keep you from getting too sick. So maybe you only feel bad for like 1 day, right? But the further into the future you go, the more that this virus is mutating, right? 3rd infection, right? Now only 2 of the antigens that you originally had immune memory against are still present. So you're sicker longer now, 4 days, 4th infection, so on, until maybe like a year later, right? A year later, the virus has mutated to the point where none of the antibodies that you had developed against it a year before are useful, right? And as a result, you're back to square one and you're sick again for 14 days and you have to come up with the selection of new antibodies to fight against it. Okay. All right. We are out of time today. There's still a few more slides. What I will do is I'll just continue with the remaining slides as the start of our next lecture on Wednesday. Okay, because then I know it's kind of disappointing. I didn't actually get into the whole COVID-19 thing, but hey, this gives you something to look forward to for Wednesday, right? We'll now talk about what happens when different viruses jump, not from person to person, right, but certain circumstances when it jumps across species, right, from other animals to other animals, and maybe other animals into us, right, which is the story of what has happened with the SARS-CoV virus, right? Okay, so we'll stop here today and we'll pick up where we left off on Wednesday.